hasn't emerged by kind of an evolutionary bifurcating event. It's a hybrid between, in this case, this lineage here and this lineage here. And so when you start to draw the evolutionary history of taxa that are hybridizing, it becomes much more like a network. And to do this, you need a lot of nuclear genes to track the evolutionary history. And up till recently, we haven't had this in ferns. Most of the work has been done by sequencing chloroplast genomes or sequencing individual chloroplast genes using the traditional PCR approach. A few nuclear genes now are becoming available. They're tricky to work with because sequence divergence in the nuclear genome is so much more unpredictable that getting primers, PCR primers, to work across a wide range of taxa is very difficult. And so recently I started using a different technique that's been around quite a while, is target sequence capture. And this is generating a normal genomic library, but it gets interrupted. What we have, if you remember, we did some transcriptome work, so we have a lot of data on the expressed genes. Of course, these are the transcripts, so they lack the introns. Fortunately, there's work on frogs and fish and even some other angiosperms that has shown that if you can design some probes, even from the exon regions, you can use that to selectively capture genomic DNA that has the introns. So we figured we scraped together a bit of money to try some target sequence capture. We designed these little oligonucleotides that are about 120 bases long. We designed them to work across different fern families, across the entire fern phylogeny. Targeted about 25 orthologs altogether. And so the process gets interrupted by these little oligonucleotides that we've designed, thousands of them, and they're attached to magnetic beads. And essentially all you're doing is pulling out the genes you want to enhance their sequences in the genomic library relative to all the other random stuff. Just got the data back last week. So we got data from 25 species. We've had enough time to go through, and most of them indeed have the 25 nuclear genes. With the process right now, because a lot of these things are polyploid, they have extra copies, so we've got to sort out the paralogs, and we're in the process of doing that right now. But from what we can see so far, this looks like a very feasible way of collecting large-scale data. The data from this, now remember, is going to be genomic DNA. We designed the initial set of probes from the transcriptomes. This should give us a cleaner data set from which we can now redesign the baits and get hopefully a much more powerful approach to grabbing these. This is a much more feasible and reduced labor, reduced error compared to PCR. If you were to try to sequence PCR amplified, say 100 genes from 1,000 taxa, it would just be too much lab work. Also designing the primers. Here, you can just build these genomic libraries relatively quickly and do this target sequence capture. So this is one of the directions I'm planning on going for the next few years. Getting the analysis, this analysis completed, and using this as the basis of probably my next NSF grant proposal. And particularly to use it to track how much effect hybridization has had on fern evolutionary history. Okay, I'm going to digress and hopefully explain why I'm suddenly changing direction. Talk about teaching. First of all, none of these ideas are mine. These are ideas that I've got from books, from reading research papers, mostly from my colleagues in STEM education and colleagues who do research on pedagogy. And I started to collaborate with colleagues on this topic about 9 to 10 years ago. And so what I'm going to do is explain some of the main principles that I use in relatively general terms when I'm designing a course and when I'm thinking about how to teach a particular topic. And it doesn't matter what the topic is. Whatever it is, always need to start with the learning objectives. What is the course? Is this freshmen or they advanced pre-med students? What skills do you want the students to learn? What concepts do you want them to understand, and what information do you want them to retain at the end of the course? And if you start with what your objectives are, it makes it a lot easier to achieve those objectives. The other thing is, like many things in science, trying to get to the solution can be best approached by just flipping the problem on its head. And so the problem here is you're trying to help students to learn. Flipping that on its head, you think about the opposite process, which is forgetting, which is something at which I think I'm an expert. And not surprisingly, the literature on this is not new. There are papers dating back to the 1880s where psychologists were studying how we forget things and how quickly we forget things and how surprised we are at how quickly we forget things. And you can plot these things called forgetting curves. The more recent cognitive science literature has shown there are some key points on these forgetting curves. And the rate at which you forget material is a function of many things, including how complex it is and how badly you want to remember it. But if you have got a piece of information that you're wanting to remember, or rather you don't want to forget, if you review that information a few minutes after you first gathered it, you're not going to be doing much in your brain because you've only just received it, you haven't begun to forget it. If you hear something and then 3 weeks later you hear it again, chances are you've already forgotten it and it's too late to do anything. There are some sweet spots that you can exploit where you or the student are in the process of forgetting. And if you interrupt that forgetting process, you can actually alter the place in the brain where the information is being stored, or so I'm told by the experts. And so when I design a course, and I've got my learning objectives and I know what the important concepts are, I try not to just kind of throw them at the student in a linear fashion. I think about, okay, this is a really important topic, so I'm going to introduce it on Tuesday. But I'm not going to go into a huge amount of detail. I'm going to set it up in a certain kind of context, and then I'm going to give it to them in fairly broad strokes. But then 1 or 2 days later, that's one of the sweet spots, I'm going to hit them with it again. This time their brains are already a little bit prepared. They've heard the information before. They're right in the process of that first level of forgetting, and now it's kind of being implanted a little bit deeper. And then a week later, I hit them again in even more detail, and that's another one of the sweet spots to that forgetting curve. So about 24 to 48 hours and about 6 days, that kind of cycle is what I aim for in a course like genetics, where the concepts are kind of that level of complexity. When I'm teaching a botany course, I've got actually a little bit more wiggle room because the concepts tend to be a little simpler. So there is a relationship between those time frames and the complexity of the information. So it isn't just repeating information, it's repeating information at that right time. The other thing is there's a lot of evidence that testing, as much as students hate being tested, testing helps. Even if it is failing. So even if a student is taking a test and just cannot remember the information, the process of just banging your head against either your fist or the drywall is actually doing something. Even if you can't remember what you had on the shopping list, trying to think about what it was on the shopping list is wrapping your brain around the problem. And as soon as your spouse reminds you a dozen eggs, click, it'll stick in that much harder. Doesn't matter what the information is, doesn't matter if it's a shopping list or a class. Information will stick if you're in the process of desperately trying hard to remember what it was. I knew he told us that, but I can't remember what it is. So, testing. And so what I do is I use clickers on a daily basis in teaching. I give them lots, rather than lots of big finals and big midterms, which I do, but I focus on a lot of short quizzes, not all of which are graded. Some of them are graded and some of them are not graded. I'll get back to that in a moment. The other is throwing the same topic in different contexts. The hardest part students have in learning, and we probably all know this, is most students, when they're learning a new topic, don't have the background context for understanding the information. And so you try and often teach both the context and the new information at the same time. One good approach to this is to approach the topic from multiple angles, but also kind of jump around a little bit, which is why I put the teaching in the middle of this lecture rather than at the end. The other advantage is that some of you were getting a little bored with genomes, that you might be interested in teaching. And likewise in class, you can retain the interest of a few more students by jumping topics around. You'll get complaints about, oh, Dr. Wolf keeps changing the subject. I'm doing that because there's evidence that that actually helps learning. The other thing I do, and I've been throwing them in here, because this is kind of a science seminar, but I do this in teaching. I don't just torture the students, I tell them why I'm torturing them. I give them citations, I post the papers, and I'm trying to help them become better learners. And the argument I present is, you can get you can get better grades by spending less time if you study efficiently, and I'm showing you how to do that. So these skills aren't just going to help you get an A in genetics, they can help you have more free time to go and play out outside and do the stuff you like, and do well in classes. Most of them ignore that, but a surprising number of them actually follow my advice, or follow the advice that I've been given. But the citations and the reasoning I find is usually useful. Okay, but this is what I hate. I'm really bad at this. So I'll give a clicker question. I'm expecting, I did a great job of the lecture. I knew I nailed it. I give them a question, and 90% of them get it wrong. And that happens, and it's probably my explanation, or it's probably the timing, something. And so the students want to know why they all got it wrong. And I'm the teacher, I want to explain, because I can see exactly, based on the answer they gave in the multiple choice, what the mistake was. And so they want to hear the answer immediately. I want to tell them, but the evidence says, no, shut up. Keep your mouth zipped, don't say anything. This is really, really difficult. The best thing to do is to sit on it for 24 hours. Let the students, most of the students will go out and forget about it and nothing will happen. But 10 to 20% of the students will be thinking, why did I get that wrong? What happened? 24 hours later, they'll have figured it out themselves. And letting students figure out themselves why they made a mistake is incredibly important for learning. Because then their own brains have solved the problem, and therefore it's been fixed in there much more rigidly than if they were just told the answer. Along those lines, learning is frustrating. It's very frustrating. Any of you go to Utah and want to learn to ski, you're not going to go from Alabama to the slopes of Snowbird and glide down effortlessly. You have to fall on your butt and hurt yourself. And I spent years in pain and frustration learning to ski, because I fall over to the left, I fall over to the right, back, forward. My brain is making these little adjustments all the time. And it's horrible, it's frustrating. I didn't have a citation here, but there's a lot of evidence that actual frustration is in itself really good. It is the learning process. There is no painless way to learn anything. And it doesn't matter if it's motor learning, motor skills, or academic learning. But you have to find a sweet spot. If it gets so frustrating, you're just not going to want to ski ever. If it's too easy, you're never going to learn. And so trying to find the balance is, for me, the hardest part of teaching. The other thing is preparing the brain for learning. Remember, the students, anyone's going to want to learn information if they've got some motivation to learn it. In the lab, this is really easy. They design their own experiments, so they're always wanting to find the answer. And in lecture, the key here is to give them a problem to solve before you've given them the tools to solve it. And so I do a lot of problem sets in my classes. Sometimes they're on their own, sometimes they're in groups. Sometimes they're graded and sometimes they're not graded. But they're problem sets for which most of them have not yet gathered the tools. So when you give them the tool, they immediately say, oh, now I can see how to use the tool. It's essentially like giving a learning carpenter a hammer before you've shown them nails and wood. Show them what the problem is first, and then give them the tools. Mistakes are good. Falling down is a good way to learn to ski. When you fall, the brain is actually doing stuff. When you make mistakes and you learn from those mistakes, you are learning. And yet our entire education system is based on punishing you for making mistakes. Okay, so there's a fundamental flaw in most of our approach to education by punishing mistakes. And so this is not an easy problem to solve because you have to have a level of assessment at some point. But in my problem sets, they are not graded based on getting the correct answer or not. They're based on the way they explain their logic. So they can give a correct answer with no explanation and get close to 0. They can get completely the wrong answer, but have a solid explanation. And at any point on these problem sets, which account, I think, for about 20% of the grade in most of my classes, if they get something wrong and then redo it, I'll give them full credit, because that's the learning process. So we constantly need to assess the students to see how they're doing. So I give a lot of clicker questions. I'll repeat clicker questions or the same concept from a different angle to test them. In the same way, the teacher needs assessing. I need to know what aspects of my teaching are working, what is failing, what should I change. And I have not been good at this. I've only recently started to collaborate with assessment experts. This is something that's becoming much more important in higher education. And those people who are experts at assessment are becoming rarer and rarer commodities. But there are techniques. It's very difficult in all situations to be able to control the experiment, but it is possible to look at games under different conditions and get some relatively quick feedback in addition to longitudinal feedback, and analyzing data from different institutions. So assessing yourself as well as a student is also important. The other thing is, in the same way as throwing students constantly into different situations, so in a 50-minute lecture, I'll lecture for about 10 or 15 minutes. They'll break up into groups to do problem sets. We'll discuss, we'll have another short lecture. I'm jumping around a lot, so they're never doing the same thing for about more than 10 or 15 minutes. The same thing, I find that putting myself in different situations is really helping my teaching. I've worked with a group teaching middle school students, which initially was one of the most terrifying experiences of my life. I thought it was. When I actually did it, they were fantastic. They were actually more creative than most of the undergraduates. So I actually have completely changed my opinion of 8th graders. But I still do some outreach to local high schools, doing some DNA experiments, DNA extraction. There's a lot of these teaching tools available. And I enjoy this bit quite a lot. I also take my graduate students out to the schools as well. not just for the fun of it, but because that whole feedback from seeing how the learners are learning can teach you a lot about teaching. Okay, changing topic again, back now to plants, but now I'm shifting a little. The first part was dealing with genome structure and evolution, and now I'm going to be focusing much more on the population level. And I've worked for some years with collaborators at Utah State University on the western aspen, which is an equating aspen, which is the most widely distributed plant in North America. It's a very important forest species. Fires in western North America are spreading rapidly through the pine forests. They get to a grove of aspen, and that's often enough to stop a high temperature fire. So there are important fire breaks, and information would be really useful on the genetic structure of these populations, looking at which genomic regions are under selection for different habitats. Remember, a very wide ecological range here. And restoration after fires, using genetic techniques to determine which seeds and seedlings to use. So about 5 years ago, I started switching over to some population genomic approaches. The whole class of these approaches, I know some of you are already using, they're called reduced representation genomic libraries. The idea being you want to sequence a little bit of the genome, but the same part of the genome from a large number of samples, so that you can get single nucleotide polymorphisms that are available from as many of your individuals as possible and get as many loci as possible. So you want to sift through the genome to find these comparable sets. So the technique, there are more acronyms out there than protocols: GBS, genotyping by sequencing, RAD tags or RAD-seq, and there doesn't appear to be a very good association between the acronym and the actual protocol. But they're all the same kind of idea. You chop the genome up with a restriction enzyme, and that hopefully gives you homologous parts in the genome across your samples. And then you excuse me, attach these little adapters that have the sticky end of the restriction enzyme. They'll have a barcode, internal barcode, unique to each individual, and then part of the Illumina primer sequence for the sequencing. You throw it at an Illumina platform and get hundreds of millions of reads. You use the barcode on that first part of the sequence to sort your reads into individuals, and then you can start screening across individuals for homologous regions and single nucleotide polymorphisms. So on the aspen, that was published 3 years ago by one of my grad students, we had over 100,000 SNPs. We were able to look at population structure, gene flow, and geographic relationships in aspen. One of my colleagues has now taken this a step further and is using it to do genome-wide association studies to look at adaptive traits for resistance to fire, resistance to disease, and resistance to drought. It's an extremely cost-effective analysis. Up until now, it's been taking a bit of time to process the data. We were developing our own software. Other people were developing their own software, and so we're kind of focusing down on a few different approaches. We do not, because every project is asking different questions, we don't rely on a single analytical pipeline. In the aspen, we found strong evidence of genetic differences between North and South populations, and now the next stage is to be focusing on this area in Montana where those two different regions come into contact and get a more fine-scale population analysis going. Another example of this type of project is another grad student of mine, R.M. Duffy, who studies these fern gametophytes. Most ferns alternate between gametophyte and sporophyte. There are a few species that are only found in nature as a gametophyte. They've lost their sporophyte phase completely. Some of these species are tropical. You find a sporophyte in the tropics, you come up into North America, in temperate regions, and you only find the Bermuda fly. Turns out northern Alabama is one of the hot spots. This species is found in several places around northern Alabama. They're found in sandstone caves. If you don't need a flashlight in the cave, then you won't find this. It's only in places dark enough where you need a flashlight. So there's a lot of interest in the physiology of these things. They weren't even recognized as ferns when people first found them because they look more like algae. But this particular species occurs throughout eastern North America, and Aaron used the RAD-seq approach, detected more than 80,000 loci. He found evidence that there are actually 3 species involved, not one. And he found evidence of based on the read depth of different alleles, he found evidence of one of the species being triploid. He was detecting multiple genotypes in sites, but he was also finding similar genotypes in very different areas, indicating that the whole population structure was much more consistent with recent long-distance dispersal, which completely goes against the initial hypotheses that these things were relicts from previous warm climates where the sporophytes were present and these species were spread throughout eastern North America. But Aaron's evidence suggests that something perhaps is moving these species around much more recently. So I have several other of these projects going on. A recently graduated PhD student was working on a mustard in the genus Bokera. This genus is known for extensive hybridization in addition to apomixis, a form of asexual reproduction. I have several students working on rare plant species, in particular western buckwheats in the genus Eriogonum. I have a colleague, Don Harbour at Loyola University in New Orleans, who's working on introductions of another aquatic fern in the same group as Azolla, the genus is Salvinia. This is found in Alabama, and it's been introduced from South America, and we're looking at populations from Florida through Georgia, all the way to Louisiana, and then comparing the variation there with variation across its home range to examine the number of origins and where in South America they were introduced from. I have a series of projects working, this is for the Army Corps. The rare plant work is funded by US Fish and Wildlife. I have projects working on wetland species. The Army Corps of Engineers does a lot of wetland plant designation work. So they need to know, is this a wetland species compared to that upland species? Are they the same species or different? Sometimes you'll see a species growing in uplands and wetlands, and yet they want to know if it can be used as a designator. And so we've got projects going on with birch and Eidenbush in western North America. The birches are up in Alaska. Looking at the genetic differences between these different species and these different ecotypes. And then finally I've got a project, another PhD student working on that fern, the sea fern, that's being used for a genome project. Similarly, when the Arabidopsis project, sequencing project started, we actually didn't know anything about the phylogeny of Arabidopsis. And it turned out for several reasons to actually be a relatively poor example to use for sequencing because it was not representative. Unfortunately, we think this is the case that the strains being used for sequencing might actually be a hybrid. And so one of my students is sampling Ceratopterus from all over the world to first figure out, and then counting chromosomes, doing some fairly traditional botanical work, and looking at the relationships and the polyploidy to find out, is there a better model, a better strain that we can be using to sequence the fern genome. So in summary, I'm interested in sequencing genomes that haven't been sequenced before, doing genome scale genotyping. Target sequence capture is the approach that I'm using for the nuclear gene analysis now across the widely divergent species. But I still use some traditional approaches, morphology and cytology. And more recently, I've started using analysis of museum data, particularly herbarium data, to look at distributions, changes in phenology, the timing of flowering in species, just using data that's already available publicly. So these are the techniques that I use in my research program, the kinds of questions I ask. What is the content, the structure, and the evolutionary patterns of particular genomes? How much do these species hybridize, and what effect does that have on genome structure and evolutionary history? How genetically distinct is this particular federally listed species? Sometimes we find that a listed species really is absolutely distinct, but we've also found in a couple of cases where it's not distinct from its very common relative. What are the origins of invasive species? How many times has this invasive species been introduced, and from where? I have another project starting this summer with a colleague in Idaho looking at some grasses from Europe. And then in general, I like to use the data to analyze population structure and understand how the range of species and changes in the range of species alter its genetic structure. I want to finish up briefly now with one component that I really try to emphasize with my students. We're taught from an early age that science should be reproducible. And yet, most of the time it's not. This is actually thought to be a fairly generous finding from a survey done by Nature, that most scientific research papers that are published cannot be reproduced. Most scientists are even unable to reproduce their own science. One of the reasons is simply that there's not enough room in a journal article to describe every painstaking detail of the protocol or the analysis. If you did that, the readers would get bored very quickly. But there are actually fairly simple solutions, and I try to convey this early on in my mentoring of both undergraduates and graduate students. The first step is, when you sample organisms, make sure that you collect voucher specimens. Doesn't matter if it's a plant or an animal. Make sure they're georeferenced. I have seen entire thesis projects totally fail because this advice was not listened, not adhered. If someone else doesn't know where to go to find those plants or animals, they can't be sure that you've got the right species, or you've got one species rather than two. It doesn't matter if you make a mistake, but you need to be set up so that someone else can find the mistake. So our methods, as I said, need to be documented in more detail. The analyses need to be publicly available, every aspect of the analyses. But again, none of this can go in the journal. It would take up too much space and be too tedious. But there are a very large number of public repositories, library repositories, journal repositories, where all this information can go. So I teach my students to build this reproducibility, and from an early stage, we use a number of different techniques. I don't advocate any one method. You can use simple text files and post these with supplementary data in digital commons or with the journal. And this particular tool is a kind of markup language that's used to put in not just the kinds of bits of software that you were using and how to get that software and all its dependencies, but what command line instructions you send to the computer to analyze the data, and then what each of the steps are. This is way too much for a journal, but now it's documented. The beauty of it is when a reviewer says, you made a mistake right at the beginning, you should have first done this, trimmed these bits of sequence off your data. You can actually go back, and you've got now a nice complete record of everything you did, and it makes it much easier to go back and redo the results. So all of this, you can even include the code that you use to make the graphics. Papers are being published now with this level of detail in the supplementary information, so that everything can be repeated on a different data set, or the same data set can be analyzed in a slightly different way. And the whole goal here for me is to make my science as user-friendly as possible, not just for other scientists, but hopefully also for the general public who, after all, are funding my research. So I think I went over time a little bit. I'll try my best to answer questions if anyone has them. So thank you very much for listening.
A couple of questions, Paul?
Yeah.
Have you ever considered flow interactions between the genome of the chloroplast versus the nucleus? Because you said, well, if I do this phylogenetic array using just the genome of the chloroplast, it will not tell an avenue of the nucleus.
Yeah.
But what if that chloroplast really has an interaction with the genome of the hybrid, for example, and that selects actually the hybrid?
So there are quite a lot of interactions going on between the chloroplast genome and the nucleus genome. But as far as we can tell in the ferns, it's almost always one way, going from the chloroplast to the nucleus. So when we look at our nuclear sequences, we will find little chunks of chloroplast DNA in there. The big thing, but it's on a relatively small scale compared at least to what's happening with the mitochondrial genome. The mitochondrial genome is, I think, the mediator for movement. The mitochondrial genome appears to be taking up this chloroplast DNA and nuclear DNA. In the hybrids, we just simply don't have enough information to know what's going on. The few cases where we've got hybrids and we've got sequence from the parents, we always see that the hybrid has all copies of the parental nuclear DNA from all the genes we study, and the chloroplast DNA of only one of them. That's what we've seen in every example. But hopefully, if we start looking at hundreds of species, we will start to, I'm sure, see some exceptions. Because you're right, there's a lot of interaction going on between these genomes. But as I said, I suspect it's more with the mitochondrial genome.
Yeah, you mentioned about target capture
stuff,
and then you mentioned, of course, the difficulty with multiplex PCR. Doing multiplex. Are you talking about getting as many copies as if you were to use PCR, or just enough to do what you need to do?
So what we're trying to sort out is that we'll target what we think is a single ortholog. And in some species there'll be one copy, and in other species there'll be multiple copies. And we're actually using that now to map the gene and genome duplications on our phylogeny. But it's just taking a little bit of extra time to sort out the orthologs. But the way in which I was describing it as cost-effective compared to PCR is simply the numbers. If you have to PCR amplify 100 genes from 1,000 plants, that's a lot of PCR. There are multiplex tools to do that, but you've also then got to be designing the primers. If the same primer would work across fern phyla, remember the ancestor of these ferns is about 200 million years older than the oldest angiosperm. So the sequence divergences we're looking at are much older than if you're working on angiosperm genes. And so designing PCR primers is a lot more challenging, whereas you can design these baits where you can design a bait that will work from one particular region. You can design 50 different baits that will work on 50 different types of ferns.
So, Paul, this is from your education piece of the slides. You talked quite a bit about testing, feedback, and assessment.
Yes.
It's something that doesn't come easily to subject matter experts.
Yeah.
So, either based on experience or from literature, do you have a sense of what the class sizes should be to provide these kinds of rapid feedback, assessment, or maybe not class sizes, but how a class should be set up for freshman biology courses, for example? Where is research?
Yeah, so I only recently found someone I consider a perfect assessment expert. I just started working with her. Of course, she's moved to UC Berkeley, but we're still going to work together. And she's been teaching me a lot about Well, first of all, the class size, there's a lot you can do with different class sizes. With the clicker questions, I'm lucky in that the maximum class size I've ever had to deal with is 150. I've got colleagues at Utah State University who teach the freshman classes where they teach 450 students. Those leave and the next 450 come in and they give the same lecture. But they still are able to use clicker questions. So the clickers can give you very quick and instant feedback. So if I'm trying to gather assessment for whether they're actually learning a concept, I can get that pretty quickly. The harder part is the more general course design approach where you need more of a longitudinal study. And that's where I'm ignorant and just starting to learn and gather information from the assessment experts. They are using information from a lot of different institutions and looking at patterns. So that's a data science approach that's being used. But this colleague is also helping me try to design some assessment tools that I can tests that I can give at the beginning of the class, at the end of the class, and do this over years to start comparing different approaches. It's always tricky because the controls are often lacking. And then if you're changing things on the fly, it makes it also difficult. But for particular concepts, rather than the overall approach, seeing if students are understanding protein synthesis or particular component in a genetics class, the clicker questions will give me quick, instant feedback within a minute if they're getting it or not. And that's useful because sometimes it's very difficult to interpret the blank stare. Does that mean I've got no idea what you're talking about, or I've heard this 100 times before on board? And the clicker question will tell you quickly. Are they getting it? We can drop the topic and move on, or they're not getting it, we need to take a step back and learn and redo it.
Thanks. At one point you said that you were interested, is there a signature of polyploidy in friend genome?
Yeah.
And I'm wondering what would be a couple characteristics that would suggest that you'd found that signature, but also wasn't a clear marker of the signature that would let you know that maybe you're on the right track, but you're not sure. Are there like a few examples of like what would suggest that you'd found that signature, but also weren't?
Well, the suggestion we've got right now is seeing these peaks in the gene duplications, which is the same signal that's been used in flowering plants and even some animal groups to detect whole genome duplication. But you're right. We're not really going to know how strong that signal is until we start getting genome structure data. And this has been done a lot, particularly in the grasses, where there's obviously you've got evidence from chromosome numbers. That's always a good clue. You've got the gene duplication distributions that are a good clue. But it isn't until you get genome sequences and good solid assemblies that you can start to see synteny across chromosomes. Once you start to see the same sequence of structural genes being repeated across a haploid genome, that's where you start to get much stronger evidence. And that's what has been used to show that a lot of this transcriptome approach for looking at the duplication peaks is actually accurate, because in those examples they've actually been able to find the signature in the genome structure. But you're right, we are basing all our evidence right now is hints. We need the genomes before we can really look at the genome structure and get that.
I want to go back to the teaching and learning issue. I was curious if you've heard about Nassim Taleb's Anti-Fragile.
No.
He wrote a book called The Black Swan, and his latest is Anti-Fragile. So it reminded me, because you talk about how students can learn if they make mistakes.
Yep.
And so his point is that by being challenged and failing in some cases, you actually get better.
Oh yeah.
But that's actually a problem, because students get extraordinarily uncomfortable if they don't think they actually know, or they get full marks or something. So it's a hard lesson to teach. So the question really is, do you have any, what do you do?
Well, as I said, that the main thing is to give them a chance to make mistakes without being penalized. And I'm not able to do that across the board. I can only do that on a small component. I'm absolutely open to suggestions and I also give them a chance to correct their mistakes. For courses where they do papers, for example, any student that throws in a bad job and doesn't cover the topic and gets a bad grade, and then asks, can I redo this? I'll be great. On the lab classes that are a bit smaller, I found that a lot easier. But I just, at the moment, my only approach, which isn't very systematic, is simply give them every opportunity to learn from their mistakes without being penalized. But if there are other practical ways to do this, I'm willing to try anything.
I'm curious to ask, what is your class average usually in your classes? Because when students, you challenge them, but you don't want your class average to be an A.
Yeah.
Because that means it was too easy, and they say an average should be a C.
Yeah, I do get so this you've touched on several very important issues here. One is the grading. I deliberately, it didn't change that much when I started to switch from the more traditional lectures. So my average is around a high C, low B. I still get reviews back complaining that this class is too difficult for the average student to get an A. And I don't.
But the average student shouldn't.
Exactly. But yeah, here is the sad thing, and I've got tenure. It doesn't matter if I get nailed in reviews, but when I started teaching like this, my reviews went down.
Wow, that's interesting.
The distribution changed. There was almost a little bit of a bimodal distribution. I'll have this little group of students that I think are getting it and seeing the benefit, and probably it's because they're applying the tools to other classes. So I always get this little clump at the top of good reviews, but my average is, I'm getting nailed. Fortunately, our upper administration is kind of aware. This has been documented across higher education that when you start to switch to more effective teaching methods, you'll often get worse reviews.
Yeah.
And I think administrators need to be aware of that. The other thing, though, that I think is important, and this gets back to the question of assessment, I think one of the most important things is peer review. I think the thing that I found helpful when I was trying to modify my genetics classes 10 years ago was when these people who studied how to teach came in, listened to me teach, and said, next time try this, do this. But we should be doing this all the time with our peers. This is, again, nothing new. This has been done at middle school, high school, even elementary school across this country and other countries. But our colleagues in the same department need to be going and critique. I mean, we need to be pretty harsh, at least as harsh on our friends and colleagues as we are on the students. Because, again, it's the only way we're going to learn and improve. But I think peer review, again, there's evidence that really helps.
I think that's great. I love that you're as interested in your student learning as you are in your own learning. But I do have another question. Your tools that you mentioned, the reduced representation, genomic libraries, and the target sequence capture, do you have publicly available pipelines for your data analysis?
Okay, so there are two. So I've got one that I developed in my lab, and it's on the GitHub repository. But don't use it, it sucks. It's like I worked with a bunch of different students, and it was also developed a little bit for an earlier approach we were using that was developed for the Corn Genome Project. But right now we've narrowed down on two approaches. One is published by, his name is Darren Eaton, and he has a pipeline called iPyRad, or PyRad. And it's not using any particular particular tool. It just puts all the tools together in a very nice pipeline. Very nice pipeline, but different from the kind of pipeline that a lot of people are using where you plug in the data, hit return and go away and let it go all the way through. His has 7 stages where you stop and you assess and you can feedback, you can do branches. I think that's the best one out there. A colleague of mine, Zach Gompert, who is one of the key developers of RADSeq, has his own pipeline, which is very similar, but it's a bit more manual, and it's nowhere near as well documented as IPyRAD. But if you've got RADSeq or GBS or any of this type of tumor type data, I'd take a look at Darren Eaton's IPyRAD. I think it's quite clean, documented, published.
Thanks everyone for taking the time.
We get a couple of volunteers to help rearrange the tables before we leave.
Yeah.